Welcome to the lecture on animal reproduction and development. After this lecture, you should be able to demonstrate an understanding of the health implications of human anatomy, physiology, and reproduction. You should be able to list the parts of the reproductive systems, explain the basic functions of the reproductive systems, explain what factor determines biological sex in humans, discuss mechanisms of sexual hormone balance in males and females, and list the leading methods of contraception and their effectiveness in preventing births and sexually transmitted diseases. Animal development begins with reproduction. Together, reproduction and development are shared features of all multicellular life. Some animals reproduce asexually. Asexual reproduction involves only one parent, whose offspring are genetically identical to the parent and to each other. Asexually reproducing animals include sponges, cnidarians like jellyfish, hydra, corals, and sea anemones, and a few others. Unfertilized eggs of some bees, aphids, and even lizards can mature into asexually produced adults. Asexual reproduction requires minimal energy, and it's advantageous in environments that don't change much over time. Sexual reproduction, however, is very common among animals. It requires two parents. Each contributes half of the DNA to the offspring. In many species, sexual reproduction entails high energy costs. And although sexually reproducing individuals use energy finding and courting mates, variation among their offspring is adaptive. It's beneficial, especially in changing environments. Haploid gametes are the sex cells involved in sexual reproduction. The gonads of sexually reproducing individuals produce haploid gametes by meiosis. In the case of humans, sperm and eggs. Gametes unite at fertilization, forming a diploid zygote. Fertilization can take place externally. This male sea urchin is releasing sperm into the water. If a sperm cell unites with an egg cell released from the female, then fertilization occurs externally. Fertilization can also take place internally, like with these birds here. Internal fertilization occurs when gametes unite inside the body of one of the parents, usually the female. Development begins with the zygote, so the zygote soon begins to divide after fertilization is complete. Soon, cells begin to differentiate or acquire specialized functions. Genes determine the overall shape and structure of the animal's body in a process called pattern formation. Development can be indirect. An animal that undergoes indirect development has an immature stage that looks different from the adult, like a caterpillar changing into a butterfly. This is indirect development. Alternatively, development can be direct. Animals undergoing direct development have an immature stage that looks like a small adult, like with these turtles here, tortoises. Which do you think humans have? Do humans have indirect or direct development? All right, let's start here with a comprehension check question. So what's the role of the zygote in the sexual life cycle? Does it produce gametes? Is it the first cell of a new offspring? Is it the first differentiated cell of a new offspring? Or it represents the last stage in reproduction before fertilization? A zygote is the first cell of a new offspring. It's a fertilized egg, an egg that's been united with a sperm. What are some of the pros and cons of internal versus external fertilization? I want you to jot down some thoughts in your notes here in your own words. Reproductive systems produce and transport gametes. Male and female systems both include paired gonads, testes in a male and ovaries in females. These are the reproductive organs that contain germ cells. Germ cells are the cells that undergo meiosis to produce gametes, sperm or eggs. Gametes are sex cells that meet at fertilization. The haploid nucleus of a sperm enters an egg cell and fuses with the haploid egg nucleus. Fertilization results in the formation of a diploid zygote. Here. Male gonads are the testes. Males have 2 testes in a sac called the scrotum. It's located outside the abdominal cavity to allow for temperature regulation. The long, folded seminiferous tubules inside the testes are where sperm develops. Sperm are secreted in semen. Mature sperm are stored in the coiled tubules of the epididymis. Sperm travel from the epididymis to a vas deferens, a duct that passes above the urinary bladder and connects with an ejaculatory duct. Seminal vesicles secrete most of the fluid that forms the semen. Sperm become activated before they leave the body. The prostate gland surrounds part of the urethra and secretes an alkaline fluid that activates the sperm to swim. Sperm travel through the ejaculatory duct to the urethra, and during ejaculation, the penis discharges semen from the body. Sperm development, or spermatogenesis, begins at puberty. Germ cells are formed in the developing fetus and stay inactive inside the testes until puberty. At puberty, spermatogenesis begins to occur and continues constantly throughout adulthood. Spermatogenesis starts with germ cells. Germ cells at the periphery of each seminiferous tubule undergo mitosis to produce primary spermatocytes. Primary spermatocytes undergo meiosis 1. They begin meiosis, forming 2 haploid secondary spermatocytes. Secondary spermatocytes undergo meiosis 2. And they continue to form 4 haploid spermatids. Spermatids mature into sperm cells, and as they complete maturation, sperm cells are released into the lumen of the seminiferous tubules. Lumen is a word we use in anatomy to refer to an empty space. Sperm are adapted for swimming and fertilizing eggs. Each mature sperm cell has a haploid nucleus, so here's the nucleus here, a long flagella, mitochondria, and a cap-like acrosome that helps it penetrate the egg cell. Hormones influence male reproductive function. GnRH regulates production of LH and FSH by the anterior pituitary. LH is luteinizing hormone. FSH is follicular stimulating hormone. These 2 hormones stimulate the testes to release testosterone inside the seminiferous tubules. That testosterone is what triggers spermatogenesis. All right, let's check your comprehension. So a sperm cell passes through multiple tubes along its journey in the male reproductive system. Which of the following comes last in the sequence? What's the last tube that sperm pass through? The testes, urethra, epididymis, vas deferens, or seminiferous tubule? Urethra. Good. All right, mastering concepts. What are the stages of spermatogenesis? Take a moment, jot down the process in your own words, or maybe even better, draw a diagram. Include meiosis 1 and meiosis 2 and the names of the different types of sperm cells that are produced after each of those phases. Let's talk about the female reproductive system now. Female gonads are the ovaries. The ovaries are located inside the body, whereas the male testes are located outside. Each ovary is surrounded by finger-like extensions of a uterine tube that leads to the uterus. Ovaries produce eggs and release them at ovulation. In females, egg production begins before birth, pauses, and resumes at sexual maturity. Every month between puberty and menopause, one oocyte bursts out of the ovary and travels to the uterus. Egg development, or oogenesis, is similar to spermatogenesis. Although the events of meiosis mirror one another, the timing is very different. Oogenesis begins with germ cells, just like spermatogenesis. The germ cells divide by mitosis before the female is born, giving rise to primary oocytes. The primary oocytes are arrested, they're paused, at meiosis prophase 1. Primary oocytes are on pause until puberty. Approximately each month after puberty begins, one primary oocyte completes meiosis 1. Each developing primary oocyte is nested in a fluid-filled follicle. So here's the oocyte, here's the follicle, or mini follicles here. Secondary oocytes rupture the follicle. Meiosis 1 produces 2 haploid cells, the large secondary oocyte, and a polar body, which is small and contains almost no cytoplasm. Eventually, the polar body decomposes, although it may divide into 2 polar bodies first. The secondary oocyte is released from the ovary into the uterine tube in an event called ovulation. Let's back up and talk about polar bodies again. All right, so mitosis happens before a baby female is born. So her germ cells divide to produce primary oocytes. They remain paused at this state of prophase 1 until female hits puberty. Then, this primary oocyte undergoes meiosis 1, which results in 2 cells. There's a secondary oocyte, which is a large cell with a great amount of cytoplasm. This secondary oocyte has lots of space to pack nutrients and all the things that a zygote will need. The polar bodies are the second cell that's produced by meiosis 1, and these are kind of just cast-offs. When these 2 cells undergo meiosis 2, this secondary oocyte will divide again and produce a single fertilized egg cell and a single egg cell that's ready to be fertilized. This is what's ovulated. The polar body leaves. So we end up with 3 polar bodies, kind of 3 cast-offs or cells that won't be used in reproduction, and 1 large fertile oocyte. All right, so secondary oocytes move to the uterus. If sperm fertilize the secondary oocyte, then it completes meiosis 2. The result is a fertilized egg and an additional polar body that continues to the uterus. If an egg cell is not fertilized, it travels to the uterus without completing meiosis. Hormones influence female reproductive function as well. So GnRH regulates production of luteinizing hormone and follicle-stimulating hormone by the anterior pituitary. It's this gland up here. LH and FSH stimulate the ovaries to release progesterone and estrogen, which target the uterine lining. Progesterone and estrogen regulate ovulation and menstruation. Hormonal fluctuations produce 2 interrelated cycles. The ovarian cycle is regulated by follicle-stimulating hormone and luteinizing hormone. The menstrual cycle is regulated by estrogen and progesterone. So let's look at these graphs here. During these different events, so these different parts of egg development, right? We start with developing follicles. We have low levels of LH and FSH. As those mature and ovulation approaches, luteinizing hormone and follicle-stimulating hormone spike. And this peak in luteinizing hormone is what triggers ovulation. As those levels fall off, the corpus luteum, which would have supported a pregnancy, begins to degenerate if a pregnancy does not occur. If a pregnancy does occur, then the corpus luteum remains active and develops, and this is what supports a pregnancy until the placenta is in place and ready to function. All right, and then down here, graph B, this represents the menstrual cycle. So we've got estrogen and progesterone here. And down here is the thickness of the endometrium. This is that nutrient-rich lining in the uterus that would support the development of a baby and a corpus luteum if in the event of fertilization. So we have estrogen peaks and drops off just before the spike in luteinizing hormone. This is what cues the development of, sorry, I'm having a moment. All right, the progesterone here, if you notice, as progesterone levels rise, so does the thickness of that endometrium lining. And then as that begins to fall and there's a second spike in estrogen, this is what cues menstruation to occur. The lining is shed and the cycle starts all over. The ovarian cycle controls oocyte maturation. The luteinizing hormone and follicle-stimulating hormone coordinate the ovarian cycle, which consists of follicle maturation and oocyte release from the ovary. Low levels of estrogen and progesterone trigger the hypothalamus to release GnRH, which results in the release of FSH and LH from the anterior pituitary. FSH causes follicle maturation. As follicle-stimulating hormone levels increase, follicles in the ovary begin to mature and release estrogen. The primary oocyte completes meiosis 1, forming a secondary oocyte. So all this happens with this peak in FSH. LH, luteinizing hormone, controls ovulation. Follicle cells release increasing levels of estrogen until a spike in estrogen triggers a rapid increase in the levels of luteinizing hormone. The LH surge causes ovulation, releasing the secondary oocyte, and transforms the ruptured follicle into a corpus luteum. The menstrual cycle prepares the uterus for pregnancy. Estrogen and progesterone signal the lining of the uterus, the endometrium, to thicken and then disintegrate. The corpus luteum secretes progesterone and estrogen, which act together to promote a thickening of the endometrium. So if pregnancy occurs, the corpus luteum secretes progesterone and estrogen, which promote a thickening of the endometrium. So instead of it being shed in menstruation, it would be thickened to support that developing zygote. Without a zygote, the cycle repeats. If a pregnancy does not occur, the corpus luteum degenerates into an inactive scar, and over the next several days, the levels of progesterone and estrogen gradually decline. The reduced levels of these hormones no longer maintain the endometrium, which exits the body through the cervix and vagina as menstrual flow. A zygote stops that cycle. If pregnancy does occur, the zygote divides in the uterine tubes and develops into a blastocyst that implants itself in the thickened endometrium. The blastocyst secretes human chorionic gonadotropin, which prevents regression of the corpus luteum. Continued progesterone production keeps the endometrium intact. This human chorionic gonadotropin, or HCG, this is the hormone that is detected in human pregnancy test. All right, if the egg is not fertilized, then lowered progesterone and estrogen levels enable GnRH to be released from where, and the FSH and LH released from where else? Good. So the GnRH is released by the hypothalamus, and FSH and LH are released from the anterior pituitary. What's the role of each part of the female productive system? Take a moment, jot down some thoughts in your own words, maybe draw a sketch. When can conception occur? Conception can occur as soon as an ovary releases an egg. Ovaries might release multiple eggs at different times, and sperm can survive for a few days inside the female reproductive tract. The fertile period begins from approximately 5 days before ovulation, so down here, through 3 or 4 days after ovulation. So you can see here is when pregnancy is most likely to occur in this window. Now, pregnancy is possible anytime after, pregnancy is possible at any time throughout the cycles here. It's less likely just before menstruation, but it's not impossible. Contraception can prevent reproduction. So abstinence is the most effective contraceptive because sperm and eggs never encounter each other. A tubal ligation in a female or a vasectomy in a male are surgical procedures which prevent the sperm and egg from meeting. Hormone contraception can also prevent reproduction. Hormone pills, patches, vaginal rings, injections, and implants contain synthetic forms of progesterone that prevent ovulation. If no egg is released, fertilization can't occur. There are many different forms of contraception. The most effective methods are surgery or an IUD, an intrauterine device. Here's a nice table that compares different methods of contraception and their mechanisms of action, advantages, disadvantages, and importantly, how effective they are. So people who practice these methods, how many of them or what % get pregnant in a year, right? So if you do nothing, couples who are regularly having intercourse, are 85% should become pregnant within a year. If you're practicing abstinence, then you can't get pregnant, right? If a male has a vasectomy, less than 1% of vasectomies fail and result in a pregnancy. The withdrawal method is not effective. 18% of couples who practice the withdrawal method are pregnant within a year. Condoms are also not foolproof in preventing pregnancy. Although something important to notice here is that condoms protect against sexually transmitted infections, whereas none of these methods do. All right, and here's some more. So there's also a cervical cap with spermicide. This ends up with about 20% of the people who use these regularly end up with pregnancies. An IUD, intrauterine device, has fewer than 1% of women who use these are pregnant within a year of use. Tubal ligation has similar results. Fertility awareness method. So this is when you don't have intercourse during fertile times, as inferred from body temperature and other cues. This requires careful record keeping, and its effectiveness is somewhere like 2 to 23% of couples who practice this end up with a pregnancy in a year. Hormonal contraception, like estrogen or progesterone, so like birth control pills or patches or injections, these prevent ovulation and implanting, and they're also relatively effective. They have a pregnancy rate of 2 to 9% in a year. They do have more side effects, however, like menstrual changes, weight gain, headaches, heart attack, blood clots, and other rare but serious complications. So this is a useful table to keep in mind. All right, so I did mention STIs. Diseases can be transmitted during sex, and contraception is not generally intended to prevent sexually transmitted disease. Contraception is intended to prevent pregnancy. The spread of sexually transmitted disease can be prevented or decreased with abstinence. If you have only one sexual partner, so if you're mutually monogamous with your partner, using condoms have a large role to play in preventing the spread of disease. And vaccination, but we only have vaccines to protect against a handful of STDs. So some common STDs are HIV and AIDS, also HPV, human papillomavirus. These can produce genital warts, but HIV and HPV can both cause different types of cancers. We do have a vaccine available to help protect against HPV, which is huge. A vaccine to prevent both STDs, but also cancer, cervical cancer. Hepatitis B. There's also bacterial. So these first ones are all viral STDs. We also have bacterial STDs like chlamydia, gonorrhea, and syphilis. There's also protists that are STDs like trichomoniasis, and fungi like a yeast infection. All right, so at this time, I'd like you to compare and contrast how 3 different birth control methods work, contraceptives. Pick any 3 that you're interested in. Jot down your thoughts in your own words. So fertilization leads to pregnancy. The zygote is a single cell that divides many times as it develops into a pre-embryo, an embryo, a fetus, and eventually a newborn baby. Sperms swim through the follicle, an outer layer that surrounds the egg cell. And it's thin and jelly-like. It's made up of proteins and carbohydrates that encase the oocyte, or the egg. Sperm squeeze between the follicle cells, and the acrosome, that outer covering of the sperm cell, bursts, spilling enzymes that digest both outer layers. And it happens quickly. The sperm fuse with the secondary oocyte. Fertilization begins when the outer membranes of one sperm cell and the secondary oocyte touch. The sperm's head releases its DNA as it enters the secondary oocyte. Meanwhile, the female cell completes meiosis. The sperm's DNA and the egg's DNA combine, completing zygote formation. At this point, the zygote can begin to divide. So about 24 hours after fertilization, the zygote divides for the first time, beginning a period of rapid mitotic cell division, or mitosis, called cleavage. This is a nice visual that shows how that might happen over a series of days. So they have the ovulated secondary oocyte. It becomes fertilized here in the fallopian tube. It turns into a zygote. It begins to divide here on day 2. It continues to divide day 3. So here it has an inner cell mass. It's a blastocyst by the time it reaches the uterus. Then it begins to implant. And by day 7, that developing blastocyst is implanted in the endometrium of the uterus. The zygote develops into a blastocyst. Oh, I see. All right. So a few days after fertilization, the zygote has developed into a fluid-filled ball of cells called a blastocyst. The cells on the inside of the blastocyst form the inner cell mass, which will develop into the embryo itself. The inner cell mass is also the source of embryonic stem cells. These are cells that are undifferentiated. They could become any kind of cell that the body will need. The blastocyst implants itself in the uterus. So around day 7 post-fertilization, the blastocyst, also called a pre-embryo during this time period, becomes embedded in the endometrium. During implantation, the blastocyst digests and obtains nutrients from that uterine lining, from the endometrium itself. Germ layers develop during gastrulation. So as the pre-embryo continues to develop, one layer of the embryonic disc becomes ectoderm, Another layer becomes endoderm. So if we look, endoderm is yellow. These will be cells on the outside of the developing fetal body. Endoderm will be on the inside. I'm wrong. Endo means internal, right? And ecto means outside. So ectoderm, endoderm, and then this layer in the middle is the mesoderm. Once those 3 cell layers are present, that completes formation of a gastrula. During the 2nd week of development, implantation is complete. The embryonic stage begins after implantation. So starting around 3 weeks after fertilization, the embryo produces structures that support its development. The chorionic villi project into the uterine lining and exchange nutrients with maternal blood. The amniotic cavity contains fluid that cushions the embryo and maintains constant temperature and pressure. The chorionic villi will eventually develop into the placenta. Four membranes support, protect, and nourish the embryo. The chorion, so here's the chorionic villi. The amnion, here. The allantois, I don't see that on this. And the yolk sac. These are evolutionary conserved structures found in all animals with amniotic eggs. The umbilical cord develops from that yolk sac, by the way. So this is a picture of fetal development at about 13.5 weeks. It's the beginning of the 2nd trimester. Organ formation takes place during the embryonic stage. For example, sex differences become apparent between weeks 7 and 9 after fertilization, when the genitals develop. The fetal stage begins at week 9. During this stage, organs become functional, bones replace cartilage, and nerves and muscles begin to coordinate their actions. Near the end of pregnancy, the digestive and respiratory systems mature, and neural connections form in the brain. Birth occurs around 38 weeks. Childbirth is a 3-step process. Labor, when the amniotic sac breaks. Hormones prompt uterine muscle contractions, and baby's head presses against and opens the cervix. Delivery is what occurs when the baby descends through the vagina. And 3, finally, the uterus expels the placenta. Childbirth illustrates positive feedback. So oxytocin is a hormone that causes muscle contractions in the uterus, which pushes the baby through the cervix. Baby's head presses on the cervix, and that stimulates sensory receptors. The hypothalamus triggers the release of more oxytocin from the posterior pituitary. So it's a feedback. The more baby's head presses, the more the hypothalamus produces more oxytocin, and the more oxytocin causes more frequent and stronger uterine contractions, which cause the whole cycle to repeat. So humans develop for 9 months before birth. A fetus born before 22 weeks is not viable, but after that time, the chance of survival increases. Birth is considered premature if it occurs before 35 weeks, or at about 8 months of gestation. All right, so when does fertilization occur? Or where does fertilization occur? Good, in the uterine tube, or the fallopian tube. Which supportive structures develop during the embryonic period, and what are their functions? Take a moment, jot down your thoughts in your own words. Maybe even draw a picture. All right, that wraps up our lecture on the reproductive system. At this point, you should be able to demonstrate an understanding of the health implications of human anatomy, physiology, and reproduction. You should be able to list the parts of reproductive systems, explain the basic functions of reproductive systems, explain what factor determines biological sex in humans, discuss mechanisms of sexual hormonal balance in males and females, and list the leading methods of contraception and their effectiveness in preventing births and STDs. And actually, I should make one footnote here about point C. This is something we discussed in Unit 2 when we were talking about genetics and chromosomes. In humans, remember, there's 2 different sex chromosomes, X and Y. Biological females usually have 2 X chromosomes. Biological males usually get an X from their mother, but a Y chromosome from their fathers. So that's the general pattern here. We discussed it in a previous unit, so I didn't include it in this lecture, but I just wanted to remind you of that bit. All right, as always, any questions you have, please make sure you jot them down and ask them. You can ask a classmate, you can ask me, you can bring it to class or office hours or shoot me an email, but please make sure you do follow up on your questions.